last level of the protein structure is called the quaternary level. It is the final 3D structure of 2 or more polypeptides. They have many macromolecules with multiple subunits within them, and there's always 1 or more amino acid within the structure. They consist of similar bonds like the tertiary one, so they have hydrogen and hydrophobic. Again, fear of water, so they do not do well in aqueous environments. An example of a quaternary is hemoglobin. So I'll do that out for you guys. Globin. The purpose of the hemoglobin is to bring oxygen into our bloodstreams, and it kind of looks like a bunch of squiggles, but always within a quaternary there are going to be sheets, the pleated sheets, which we saw in the secondary structure, and the alpha helixes, which we also saw in the secondary structure. So here they're just basically intermingling within each other. Here we go. And you got one up here. So basically you cut this in half and you have your alpha up here and your betas down here. They're located between the R group, so you can't see it, but if you looked in closer, they're located within the R groups. And like I said before, they're very delicate, they're not strong, and they have weak bonds. And that's basically the quaternary level. So overall, you have the primary, which is the strongest, and it has covalent bonds, which means it's even stronger, does well in aqueous environments. The secondary structure, also very strong, but they have hydrogen bonds. But since there's so many of them, they do well in a pack in aqueous environments. Then we have our third level, which is tertiary. It becomes a little more delicate. This is where the 3D shapes form, and they consist of hydrogen, ionic, hydrophobic, and disulfide bridges. And then lastly, we have our quaternary, which is similar to the tertiary, and it's delicate. It has hydrogen and hydrophobic bonds. This is an example of hemoglobin, and that is all the protein structure.